What an honor it is to be here today and be speaking ahead of Dr. Ray Weil. As a former professor, always appreciated his textbook, and a little bit, put on the spot here, make sure I got all my facts straight about soil science. So what an honor it is to be here. Thank you. So today I want to talk a little bit about soil biology and linking soil health and ecosystem function specifically to the biology that drives numerous functions in soil. And really throughout this conference, soil biology has been a resounding theme. Every single talk I went to mentioned the importance of soil biology, soil microorganisms. Who are they, what are they doing, and how are they influencing soil health and overall crop productivity? So hopefully by the end of this, you'll get a little bit better appreciation, although I have to say that listening to the previous two talks, there'll be a little bit of repetitions, and that's probably a good thing. It reinforces some of these really important key concepts. So I always like to start off defining soil health. You've probably seen this a number of times, but just to make sure, we have the continued capacity of a soil to function as a vital living ecosystem that sustains plants, animals, and humans. And I highlighted function and vital living ecosystem, primarily because what's largely been missing from a lot of soil health and soil quality assessments is that microbial piece, is the biology that drives pretty much everything that's going on in the soil. And so there's a nice publication by the Soil Science Society of America, Soils, the Foundation of Human and Environmental Health. There's a lot of parallels these days between our gut microbiome, animal microbiomes, and the microbiomes in the soil. And so what are those linkages as we produce these foods under different production systems and creating healthier soils? How does that influence our microflora in our cells? And then I also underlined function, because it's truly the functions of the soil that we're trying to address and to optimize. And I like this little cartoon here. The microbes can help feed the world. And again, hopefully by the end of this, you'll get a little bit of appreciation of why that's true. And so I like to just start out by acknowledging how diverse and abundant the life below ground is. And it's this diversity, and there's different numbers, and per 10 square feet of soil, we have 100 trillion to 1 quadrillion bacteria and archaea that are in existence, up to 10 trillion actinobacteria, they're specialized bacteria. It's hard to measure fungi in numbers, so we tend to measure their hyphal lengths, and so we can have up to 10 million per 3 foot, 10 billion algae, 10 million to 100 billion protists, 100,000 to 10 million nematodes, and they're not always the bad guys causing disease, but really critically important for population regulation and nitrogen mineralization. Up to 1 million mites, 1 million columbella, these little springtails that exist, 10 to 100 earthworms, and up to 10,000 other fauna. And if you add them all up on a biomass basis, you get anywhere, 1 acre can inhabit anywhere from 10,000 to 30,000 pounds of below ground biomass. It's equivalent, depending on the cow, anywhere from 20 to 30 cows per acre mass below ground supports all of the life that we rely on above ground. And one of the things I always like to point out when I talk about soil biology is it really takes a village, right, to make soil function. It's not just one organism, it's not just mycorrhizal fungi, it's not just nitrogen-fixing bacteria, it's not just earthworms, it's the whole collective community that works and interacts together to support all these important soil functions. So with the sunlight and a little bit of precipitation, plant roots then, as we learned in the previous two talks, help support this diverse population of microbes and soil fauna. And they all interact again together to support numerous soil functions, including nutrient cycling. We saw just previously about the importance for mining certain nutrients from the soil, nitrogen, phosphorus, and a lot of the micronutrients as well, due to the actions of fungus and bacteria. Also, I'll show you in a minute, but aggregation is primarily a biological process. We have organic matter that's formed really on the backs of dead microbes, and I'll share with you some information about that. And again, we saw, and I'll have some repeated slides from the previous talk about the importance of microorganisms in protecting those plant roots, and not only protecting, but actually enhancing their growth and overall quality and yield. And lastly, of course, we need them to detoxify the pollutants in our soils, including the conversion of agrochemicals into less toxic forms. So all of these functions work together then to really create an optimized system where plant productivity, yield, and quality are optimized. And so I have 2 little girls at home, and so we, a lot of nighttime stories, but in our house the stories are a little bit different, where we talk a lot about soil. And I refer to soil microbes as the Goldilocks in the soil. They have a very narrow range. Microorganisms in general are extremely diverse. They can be found in the most harshest environments on Earth, in Antarctica, deep thermal ocean vents, the Atacama Desert. You name it, you can find microorganisms. But in agricultural systems, they tend to be optimized in a more narrow range, where it's not too hot, not too dry, not too cold, not too wet, not too acidic, not too alkaline, just right. And so I kind of think about that, and how do we optimize these environmental conditions through our management strategies to get these optimal microbial biomass and activity that is translated under those just right conditions. However, most organisms, especially microbes, exist in a resting phase. Up to 90% of bacteria can just be in kind of a chill state, just waiting for those conditions to improve and to become just right in soils. However, so they're kind of like the Sleeping Beauty, just waiting, waiting, waiting. But other organisms can actually act like they're Prince Charming and awaken them. And that was alluded to in the previous talk with earthworms. But other soil fauna, these transporters and stimulants of other microbes, the population regulation through fauna like mites and centipedes and millipedes, and that shredding action that stimulates the decomposition of residues, can awaken the soil microbes and act as their Prince Charming. However, there are biological hotspots, these areas in soils that are really functioning and supporting enormous amounts of microorganisms. And so what we need to consider is, how do we optimize not only these conditions through management, but how can we use management to act as our Prince Charming and awaken those workers, those miners, those plant protectors that we heard in Keith's talk. So there are about 5 areas in the soil where biological activity is 10 to sometimes 100 times as high as in the bulk soil. And it really starts with the rhizosphere. This is that zone right around the roots, where, again, we've heard a lot about carbon dioxide and photosynthesis. And another thing we talk a lot about with my children, they're just learning about photosynthesis in school, and CO2 and sunlight comes in, and plants make sugars and release oxygen, and we breathe, and that's awesome. And then I have to stop them and say, but wait, there's more. Let me tell you about this. And did you know about all of the carbon that then comes out of the roots and stimulates and drives that life that mommy studies and works with, and that biology, the microbiology, then drives all of these really key functions that help support all those plant growths. So they roll their eyes, but they bring it back to their class, and they're teaching other kids about the importance of photosynthesis beyond the standard equation that we learn in schools. And these plants then, their residues are left at the surface and create a litter layer. This is especially easy to see in a forested system, right? As leaves fall to the floor and they build up and create a litter layer and go through various stages of decomposition. That's equally important in agricultural systems, as we've seen through no-till operations and cover crop operations, and leaving that residue on the surface. This is extremely important to protect the soil from temperature fluctuations, to keep that moisture trapped in the soil, to not lose it as readily to evaporation if it's otherwise bare and exposed. And not only that, but it serves as that carbon source that helps feed the life below ground. So critters come up and take this residue and then translocate it and change it into usable forms below ground to stimulate this microbial population that then is doing all those things of plant protection and nutrient cycling, plant growth, and other key functions. Key to some of this litter decomposition and movement from the surface to below ground, as well as acting as microbial biological highways, as Keith mentioned in the previous talk again, are these root or worm channels, right? So these are biological hotspots as the earthworm moves through and leaves that emittance or waste products behind. They're really stimulating that microbial population that then feeds. They're also moving microbes from the top down to different areas into the soil. And that area, that biopore, if you will, that's left behind is extremely nutrient rich and dense in microbes. And so there's orders of magnitude higher biological activity there. And then that's also a conduit, right? A highway for water and air exchange in the soil. And so where do plant roots tend to follow? They often tend to follow those worm channels because they're after those nutrients or after the water that's more easily exchanging in those root channels. So they're very, very important for a number of different functions. And when we till, right, we're disrupting those channels, those big biopores, we're disrupting water flow, we're disrupting that distribution in that biological hotspot that's stimulating nutrient cycling within these pores. And then we have aggregate surfaces, right, those important aggregates that are held together through different microbial glues and the enmeshment of plant roots. These are also biological hotspots. It is the ultimate home of the microorganism. And aggregates, of course, are made up of organic matter and those minerals in soils, but they're also creating pore space. The pore space in soils, the voids, if you will, are extremely important. They're considered the lungs and circulatory system of the soil, where water, nutrients, air exchange, and also microorganisms and other fauna. Here's a little springtail that got captured on image. Actually using one of those proscopes that Keith mentioned. So these are the biological hotspots. Where we have these hotspots, we have more efficient nutrient cycling. We have those releases of those chemicals that are helping to protect plants. How do we optimize these? How do we maximize these biological hotspots so we're not just out in a desert waiting for something to happen? Bare ground is a desert, right? There's not a lot happening. Sure, you can find microbes there and they might be active, but what we really want to do is maximize all of these biological hotspots or zones of maximum activity to maximize all of those great benefits that they're working so hard to do. And to do that, again, it's that flow of carbon through the system, and I'm sure Ray will dive a lot deeper into carbon and organic matter. But again, right, it's the plants taking carbon dioxide out of the atmosphere, converting it into sugars that are exuded and other chemicals that stimulate that microbial population. The residues at the surface then can form particulate organic matter that then is broken down, and it stimulates the microbes below ground, and ultimately come to find out that the majority of organic matter is actually built on necromass, or dead microbial cells, and plays a very key role in the production and the formation of soil organic matter. And then while they're doing this and building organic matter and all the amazing benefits that come from it, they're also helping to stimulate plant growth and productivity. And so this is a slide showing soil aggregation as a biological process. Again, there's plant roots that physically enmesh the soil into aggregates, but it's also driven by the actions of bacteria that release these sticky substances. This is a bacterial cell in that little ooze that's coming out of it is a sticky polysaccharide that literally helps bind particles together. Actinobacteria also form these filaments that look like fungi, but it's actually bridging two minerals together and holding it in place. And of course, the mycorrhizal fungi and its extensive hyphal network really creates these very strong aggregates that are stabilized at the micro scale. And these aggregates are extremely important to resist erosion, water, and wind. Again, it's the ultimate home of the microorganisms, and it creates that space for water, air, and nutrients to flow through the system, as well as transport microbes and other soil organisms from one part to another. And they're created, these pores are all different sizes. There's big pores, giant pores, medium-sized, little pores, micropores, and they all work together to do different functions. The large pores are those ones that are really important for infiltration and getting water as it hits the surface of the soil into the soil and not running off. It's important for drainage and aeration because those big pores are the ones that then drain the first, creating more oxygen flow so we don't go anaerobic and cause more problems. The smaller pores then are important for water storage and protection of that organic matter, as well as the microorganisms, because they can hide in these little nooks and crannies and evade predation. So it's really important to build these systems, create the plants that feed the microbes, that create the sticky substances and the physical enmeshment of the soil particles to hold together so we don't lose our topsoil. We get water into the ground, we don't lose chemicals to run off, and we make sure that our soils are also aerated following a heavy rain or an irrigation event. So this is from the textbook, Dr. Well. An ideal soil, right, is made up about half of that space is occupied by minerals, and then half is just space. And in that space, 45%, see, look, I already failed. 45%, I got my colors backwards. 45% is the minerals, 5% is organic matter in this, idyllic kind of soil. Of course, there is, no ideal soil. It depends on your environment, but this is kind of a good starting point. Anywhere from, 2 to 5% is considered a decent organic matter level. And then that other 50% is just space. And that space can be occupied by either air or water, and that changes depending on the conditions in the soil. So this creates a good environment. There's enough organic matter, there's enough porosity to help with infiltration and drainage. But what happens when we till or when we compact the soil? We change the dynamics of this. Now we no longer have half space and half minerals and organic matter, but the majority of it now is occupied, right? If we have the volume and we just squish more minerals into the given space, we're losing the pore space. We're losing the ability of that soil the water to infiltrate into the soil and drain, and we're increasing then our ability to lose water, soil, and chemicals. And those are dollars running off the field, not just from an environmental, but also an economic perspective. And if we're ponding, then plants are drowning. They're not getting it off to a good start, and the yield is typically negatively impacted. It also increases our susceptibility to diseases, as well as we're creating more chances for anaerobic space. Also, not only do biology drive the aggregation and all those important processes I just described, but they're also incredibly important for nutrient cycling. So the majority of fertilizer, no matter what initial form, goes through the microbes before the plant gets it. That's primarily, right, because of those trillion to quadrillion bacteria, right? They're swarming. There's more of them than there are roots. To access some of those, the fertilizer or the organic material that they're decomposing. And it can contribute to significant amounts in the soil. So the microbial biomass constitutes anywhere from 1 to 5% of organic carbon, 2 to 6% of organic nitrogen, about 3% of organic phosphorus in our agricultural soils. And that number can increase significantly up to 24% in grassland soils. I guess, I would argue that by changing our management strategies, increasing diversity, increasing cover crops, increasing those biological hotspots, that even this number would potentially be increased overall. As Keith mentioned, the mycorrhizal fungi can release cool enzymes and acids to help mine and strip the phosphorus that's otherwise bound to the mineral surfaces. And here's an example from the textbook that where microbes release phosphorus from the minerals in an incubation period of 12 days. This is how much is released in the control where there's no fungal culture that's included, and this is the amount that's released when we add fungi. So the presence of the fungi, again, through their really unique enzymatic processes and acids that they can release, are very critical in stripping and mining phosphorus. That's important. Phosphorus is one of the reasons why the ideal soil pH is that 6.5 to 7.5. It's that narrow window where phosphorus is available. But nature has evolved over many millions of years to access that phosphorus that's bound either to calcium or magnesium in our alkaline, maybe western soils, or under different mineralogy, under acidic conditions, through the actions of fungi. And also bacteria also have really specialized enzymes that can mine that phosphorus and take it out of its unavailable form and convert it into organic and inorganic forms for plant growth. So by maximizing those biological hotspots that help stimulate the fungal and the bacteria that can do this, we can increase the amount of phosphorus that's trapped in the soils. You've seen this. I love this slide. I love this concept of the communication that happens below and above ground through interactions that scientists are really just beginning to tap into. And so just to go through it again, just because it's just so cool that the aphids up here are attacking the leaf, and they're sending, the plant then sends these chemical signals, those volatile organic chemicals, into the atmosphere. That's like an SOS signal to, hopefully, if there's aphid hunting wasps that are nearby, they receive this signal that's floating in the air, and they can come in and they'll start attacking those aphids. But the plants that support these wasps have to be present for these guys to get the signal. So if it's too far away, they're not going to get it, right? So how can we manage our borders? How can we manage our field edges? If we're on pivot, what about the corners? How can we, manage these other areas in our soils, in our system on the landscape, to make sure that we have a habitat for these and other beneficial insects that can come in and help hear and sense these chemical signals, these SOS signals that are being released. You can also have blight and other pathogens causing leaf tissue disease, and then what happens down here is that the plants are releasing a chemical that stimulates a biological bacterial population that comes in and then will swarm the roots and act like a barrier that then won't allow any other pests to come in and attack that plant while it's in a sort of a stressed state. And then as Keith mentioned, there's also then these mycorrhizal connections that if this plant's being attacked, it sends a signal through the hyphae to the other plants, and it tells that plant, hey, I'm being attacked. There's the enemies in the vicinity. Shut down and build up your defense mechanisms. And then lastly, there's a lot of evidence that drought and stress can induce these specific bacteria and fungi that then help the plant deal with those stresses. So how can we choose plants that help stimulate specific microbial organisms that can then come in and help a plant through those more stressful environmental times as well. So there's this whole communication that's happening, and depending on the plant, it's releasing different exudates, and depending how that plant is managed, it changes which community it's supporting. So how do we manage for biological hotspots? The idea is to go from something like this, where we have a single crop with fallow period left exposed year after year. This is cotton, so very low residue, even if You could no-till cotton without any other crop in rotation. This is not leaving a lot of residue behind. There's a long fallow period, and we certainly want to, change from this highly disturbance process and just losing soil to the atmosphere, creating the destruction of those aggregates, that microbial home. How do we go from management strategies like this that are just very disruptive and destructive to those biological hotspots that are working hard for us if we let them, to something more like this, where we have cover, we have residue, we have diversity, we have cover crops and perennial systems. How do we maximize that diversity as well, or pastures to then incorporate animals into the system to stimulate that biology and get a jump start oftentimes on those biological functions. This is what we want. When we have this, we stimulate all those biological hotspots. We are the Prince Charmings of the microbial community. We do reawaken them, and then they come out and they're activated, they get the carbon that they need, they get the nutrients that they need, and then they pay back to the plants and provide a lot of key functions. So at NRCS, we have the 4 soil health principles. And you've seen these, but I'll just read them again. The minimize disturbance, maximize soil cover, maximize a continuous living root, and maximize biodiversity. And I tend to simplify things even a little bit more, and really what it comes down to is protect and feed. Right? So this side of the circle, minimize disturbance and maximize soil cover, we're protecting the soil, we're protecting that aggregate, that microbial habitat, we're protecting organic matter that's imparting so many key benefits to the soil system. This is protection. And on this side, by maximizing continuous living root, by maximizing plant and animal biodiversity, that can come in the form of cattle, other livestock. It also can include those wasp and predatory beneficial insects that are also important. This is where we feed and we fuel that soil biology. So if you can just think about your management strategies and anything you do, if you're protecting and you're feeding, then you're probably on a good path. So why do we want to minimize disturbance and maximize soil cover? I feel like I can probably skip over this because it's at the end of the 2-day conference. But right, we want to maintain those stable aggregates that are so important to resist erosion and keep our chemicals and our nutrients and our microbes and our soil, our topsoil that's so valuable, in place. It also buffers temperature, it reduces evaporation, and it maintains that soil organic matter. Depending on what we're doing here, our practices, we might just be in maintenance mode. We might not be building organic matter if we're just leaving residue on the surface and minimizing disturbance. But that maintenance is step one. On the left-hand side, this is where we're feeding, we're fueling, we're actually adding something back to the soil. So we're maximizing that biodiversity and living roots. This is extremely important to break disease and pest cycles, to stimulate or change that below-ground diversity. We're increasing organic matter, we're increasing nutrient cycling, we're enhancing plant growth, and we're increasing that predator and pollinator populations. So there are a number of different practices. Some of them are listed here that kind of align with the 4 principles. And you'll notice that cover crop hits 3 out of the 4 principles, and if we don't till, then it could easily be included in the 4th one. But cover crops are a great way to try to accelerate and to hit all of these key principles that then translates into those biological hotspots, that biology that's creating the functions, that's doing all that hard work to create optimal plant growth and productivity and yield and quality. So this is from the Global Soil Biodiversity Atlas. It's a great resource. It has amazing pictures of microorganisms. I've stolen a lot of these images from this atlas. It's freely available online, so I encourage you to check that out. It was just released here a year or 2 ago. And they have this kind of nice depiction where they have no-till on the left and conventional tillage on the right, and the size of these boxes kind of are an indication of the size of the abundance of that particular biological group. So no-till, right, with the protection of plant residues, protection of aggregates and resident organic matter, stimulates and increases fungi, nematode feeding fungi, or fungal feeding nematodes, microarthropods that also can feed on fungi. Earthworms are protected, their channels and their habitat is allowed to go on. And then in conventional tillage, this is where you can compare. We have a reduction in fungi and those critters that then feed on the fungi. And again, these are really important for stimulating nutrient cycling. And we get an increase then of bacteria and protists, also important for nutrient cycling, but we get a shift in the population. And we also get a reduction in overall biomass, right, when we till compared to no-till, as well as an overall reduction in diversity and an evenness that's distributed across the populations. And so these dynamics translates into a variety of different functions that are key. So one way to increase those biological hotspots, increase organic carbon, is to increase crop diversity. So this is a study at a University of New Hampshire where they looked at high diversity systems compared to monoculture. And you can see that there's higher, this is depicting the microbial biomass. It's stimulating that microbial biomass, and these residues are feeding and fueling the microbes. And we actually get an increase in organic carbon from 8 milligrams per gram to 10, which is a significant increase in organic carbon. It takes a long time often to increase those levels. They also looked at aggregate stability, and so they have these field moist aggregates and then a total amount of water stable aggregates. These are the ones that are stabilized. These are the ones that are important for resisting erosion. And then you have different monoculture, kind of simple, maybe a soy corn rotation, or corn with one cover, and then we get more complexity with soy, wheat, corn with one or two covers. And we see as we increase crop diversity, we increase that water stable aggregate, we increase the microbial biomass that's creating the aggregates that are stabilized, and we're also increasing that microbial biomass that's directly tied to that increase in organic carbon. So increasing the diversity increases biomass, organic carbon, as well as those key aggregates that are important for resisting erosion. This is a really neat study out of ARS in Colorado, where they did a nitrogen rate study, and they looked at the root exudate. So each bar here is a different kind of root exudate that's listed over here. And it doesn't really matter, exactly the composition, but note how the bars change, the amount of the residues change as we increase nitrogen content. And so nitrogen rate actually changed both the amount and the composition of root exudates. And in this case, this was an example of where more is not necessarily better, even from a microbial perspective, when translated into plant growth. Because what they found was that as is typically found in nitrogen rate studies, where we increase nitrogen rate, we tend to decrease nitrogen use efficiency, and we actually had the fertilizer is being lost to the microbes. So, right, we're increasing these root exudates. What are they doing? That's stimulating the microbial biomass. Those quadrillion, million, add lots of zeros to the end of that, are stimulating the microbial biomass. Who gets the fertilizer first? Microbes. Right? And so we actually, because of that increased microbial biomass in competition with the plant roots, they actually saw measured a reduction in nitrogen use efficiency that they tied back to that microbial community. So we want to be in that sweet spot, but just right, not too much, not too less, just right. I often get a question, how does fertilization affect soil microbial biomass? And so this was a study out of California with Kate Scow and Dan Geisler. I'm not sure how to say his name. But in general, what they found in their kind of meta-analysis was that fertilizer increased soil organic carbon content 13%, actually increased microbial biomass. And that would make sense, right? If we're increasing plant productivity, we're going to stimulate root growth and we're going to stimulate the microbial population from a biomass perspective. Urea and anhydrous do have a negative, but it tended to be short term. But what we don't know, maybe we're asking the wrong question, is what is the effects on that community composition, right? And oftentimes these fertilizer concentrations are too high for symbiosis to work most efficiently. And so that just because microbial biomass and the overall abundance isn't being negatively affected, could still have some negative impacts. And that has been translated into these studies, a little bit still controversial, but makes you pause and think about it, that the historical nitrogen additions have actually been shown to reduce the efficiency of how much nitrogen can be fixed by nitrogen-fixing bacteria. So that not just when we add nitrogen to the soil, they kind of shut down and they don't work. This was actually showing that their efficiency, taking the bacteria out of the soil and growing them in a pure environment, that how much nitrogen they fixed from the atmosphere was actually reduced. Kind of got lazy, is how they described it. And fertilization also reduces mycorrhizal fungi, 15% with nitrogen, 32% reduction with phosphorus in this study. And then, of course, Rob showed some data and talked about how cover crops can also be used as nutrient traps. And this is out of Colorado as well. But the timing of the planting influenced how much was reduced and the type of the cover crop. And then this is just another example of how different cover crop species, in this case, support different fungal functional groups. So this is out of North Dakota, and they found, these bars again are different types of fungi that they're putting into different functional groups, parasites, arbuscular mycorrhizal. They found that the spring wheat had the highest diversity, kind of more even distribution of these bars, that the AMF was highest in vetch and clover, and that oats were good for saprotrophes. Those are the ones that break down dead organic material and important for decomposition and nutrient cycling. So how do we couple this information? How do we couple this with our nutrient management, our water management? And that's all influencing the root exudates that then changes the composition of that microbial population that then could have, again, important functions in the soil. And then this is, even in vegetable systems, this is also out of California, where they looked at the influence of cover cropping compared to composting. And they found that the frequent cover cropping improves soil food web more than just compost alone. And that translated into increased microbial biomass and a higher proportion of that biomass in the organic carbon content. And that translated then to vegetable yields that were higher in those cover crop systems compared to just compost alone. So compost good, compost with cover crop even better was the kind of the take home. And that's a really challenging area, right, to get cover crops in, and a lot of pros and cons that you have to think about to try to incorporate it into vegetables, but they're finding some good results. And then this is just another study out of Colorado where they looked at different cover crop species. And in this particular case, the one that won was the sorghum Sudan cover crop. It increased the microbial biomass. It actually changed the composition of that microbiome. The potatoes had the lowest disease index here. And here they are showing the quality of that. Highest siderophore, that's a chemical that can grab onto certain micronutrients and help with plant productivity. It increased yields 12 to 30%. The quality was 40%, or 40% of the tubers were 8 ounces or larger in that cover crop, and it translated into a net benefit of $60 to $400 per acre gain because of that yield and quality improvement. So which cover crops can translate into this plant protection because the disease was reduced? How can it influence nutrient uptake, and how does that then translate? Which ones under which systems, under which environmental conditions? And then I'll just end with this slide where, this is a cover crop conference. And I guess this is a cover crop seed field. And this particular producer was out every day for 5 days straight on the same piece of land. And these are on silt loams. They're very susceptible to erosion. And I guess what they were doing was grading the land to create this drainage that then, supposedly drains into a stormwater system here to get water off the field. But you can see, how well that worked as the ponding of the soil happens and this field floods every year. So how do we work with the seed producers to make sure that the seed, we're not just translating one problem in one area for another, right? So we need to work with seed companies and find out what their challenges are, what drives them, to do this, and how can we perhaps alter those practices so that we have a full cycle of our cover crop seed that's being produced is actually produced under those same soil health management strategies that we're using that cover crop seed in other parts of the country. And so I just said all this, so I'm just going to skip it. But I want to end also with, so that might mean that we have to change our perspective. We might need to actually get our heads in the ground and look below the ground. And, this is my daughter actually listening to the soil. And she stayed there, didn't sleep, but she stayed there for about 3 minutes just listening. And we let her because we thought that was important. And I love these two quotes, right? Whether you think you can or you think you can't, you're right. And, in Master Yoda language, that's do or do not do. There is no try. If you're going to do something, do it right and give it your all. And if you fail, keep trying.